Imagine a survival system so perfectly tuned by evolution that it keeps you alive, yet so fragile that a single modern molecule can turn it into an internal dictatorship. For hundreds of thousands of years, your brain relied on a strict chemical currency to dictate what mattered, finding food, avoiding predators, and securing a mate. This currency is dopamine, synthesized in the depths of your midbrain. It was never designed to deliver pleasure. Its evolutionary purpose is to act as a precision prediction error signal. When you experience something unexpectedly beneficial for your survival, a burst of dopamine fires, stamping the surrounding environmental cues into your memory. It tells your brain, whatever just happened, remember every detail because we must repeat it to stay alive. But this ancient survival machinery possesses a fatal flaw. It cannot differentiate between a behavior that saves your life and a synthetic chemical that mimics that saving grace. When a highly addictive substance or compulsive behavior floods this system, it does not just release a normal biological pulse of dopamine. It unleashes a chemical tsunami that completely dwarfs any natural reward. The brain interprets this unprecedented surge not as a recreational high, but as the single most critical survival event it has ever encountered. It flags the substance as more vital than water, more vital than food, and more important than life itself. This is the origin of the dopamine illusion, a profound evolutionary error where the brain becomes biologically convinced that its own destruction is the key to its survival. To understand how this illusion takes hold, we must journey into the fundamental hardware of the human reward system, the mesolimbic pathway. This neural highway begins deep in the brainstem, within a dense cluster of neurons known as the ventral tegmental area, or VTA. The VTA acts as the primary manufacturing plant and distribution hub for dopamine. When triggered, the neurons within the VTA fire action potentials down their long axons, projecting directly into a structure buried beneath the cortex called the nucleus accumbens. The nucleus accumbens is your brain's valuation center. Think of it as a biological calculator that constantly computes the motivational salience of your environment. It takes the dopamine signals arriving from the VTA and translates them into an internal drive, essentially deciding how much physical and mental energy you should expend to pursue a specific goal. In a healthy state, this VTA to nucleus accumbens circuit operates within strict homeostatic boundaries. Eating a meal might increase dopamine levels in the accumbens by 50%, intimacy might raise it by 100%. However, when an addictive substance enters the equation, it bypasses the normal regulatory checks and balances. By directly forcing the VTA to fire uncontrollably, or by blocking the reuptake mechanisms that clean up dopamine from the synaptic cleft, substances can rocket dopamine levels by 500 or even 1,000%. This massive, unnatural spike completely overwhelms the nucleus accumbens, radically altering its structural perception of reality. Let us zoom in closer, down to the submicroscopic level of a single synapse within the nucleus accumbens, to see exactly how this chemical hijacking occurs. Under normal conditions, a presynaptic neuron from the VTA releases a controlled number of dopamine molecules into the synaptic gap. These molecules float across the fluid-filled void and bind to dopamine receptors on the postsynaptic neuron, sending a clear temporary signal. Once the message is delivered, specialized vacuum cleaners called dopamine transporters pull the remaining molecules back into the presynaptic cell for recycling. This maintains clean, crisp signaling. Addictive mechanisms completely disrupt this elegant recycling process. Cocaine, for instance, physically plugs these dopamine transporters. Imagine jamming a stick into a vacuum hose. The dopamine cannot be cleared, so it accumulates continuously in the synaptic gap, repeatedly slamming into the postsynaptic receptors over and over again. Amphetamines go a step further. They not only block the transporters, but they actually reverse them, forcing the cell to spew out its internal stores of dopamine into the synapse. Other substances, like opioids, work by turning off the brain's internal brakes. They silence inhibitory GABAergic interneurons in the VTA, neurons whose sole job is to keep dopamine firing under control. With the brakes cut, the VTA fires relentlessly. The result across all addictions is an extended, inescapable bath of dopamine that the synapse was never structurally designed to handle. To comprehend the long-term devastation of this synaptic flood, we must distinguish between the two distinct modes of dopamine signaling, tonic and phasic. Tonic dopamine is the steady background hum of chemical current that constantly flows through your brain. It sets your baseline motivation, your energy levels, and your general sense of engagement with the world. Phasic dopamine, on the other hand, consists of rapid, intense spikes that occur in direct response to unexpected rewards or highly salient environmental cues. It is the exclamation point in your brain's internal dialogue. In a healthy brain, there is a beautifully balanced relationship between the background tonic hum and the sharp phasic spikes. The spikes stand out clearly against the quiet background, allowing you to learn cleanly from your environment. But when addiction introduces massive, repeated, artificial spikes, this delicate balance is completely shattered. The continuous presence of excess dopamine elevates the background tonic levels to an artificial high, while simultaneously exhausting the physical reserves of the neurotransmitter. Because the background hum is now incredibly loud and the neurotransmitter pools are depleted, subsequent natural rewards can no longer produce a phasic spike high enough to be noticed. The exclamation points are swallowed by the noise. The brain loses its capacity to detect subtle, meaningful differences in value, rendering everyday joys entirely invisible to the reward circuit. The human brain is governed by a fundamental, unyielding law, homeostasis. When any biological system is pushed too far in one direction, it fights back with equal force to restore equilibrium. When the postsynaptic receptors in the nucleus accumbens are subjected to a continuous, unnatural bombardment of dopamine, they perceive this as an existential threat to their cellular integrity. Overstimulation can cause excitotoxicity and cell death. To survive, the postsynaptic neuron initiates a defensive retreat known as receptor downregulation. Through a process called endocytosis, the cell physically pulls its own dopamine receptors inside its membrane, swallowing them up so they are no longer exposed to the synaptic gap. It is the neurological equivalent of putting on heavy ear defenders in a deafeningly loud room. Furthermore, the brain alters its internal signaling cascades, making the remaining receptors significantly less sensitive to dopamine. Now, consider what happens when the addictive substance wears off and leaves the system. The synaptic gap empties out, but the neuron is left with only a fraction of its normal receptors, and those that remain are deeply desensitized. The natural, everyday levels of dopamine are now completely insufficient to stimulate this depleted landscape. The baseline state of the individual drops into a profound deficit, a state of biological bankruptcy where the normal world feels entirely flat, gray, and devoid of meaning. With the receptors heavily downregulated, a profound neurological shift occurs in how the brain processes learning and anticipation, the subversion of the reward prediction error. Discovered by neuroscientists studying learning algorithms, the reward prediction error is a mathematical calculation performed by dopamine neurons. When you expect a reward and get it, dopamine fires normally. If you get more than expected, dopamine spikes. If you get less than expected, dopamine drops below baseline, a signal known as a negative prediction error, which feels like disappointment. Addictive substances break this mathematical equation. Because of their direct chemical action on the brain's hardware, they cause a massive dopamine spike every single time they are consumed, regardless of expectation. The drug never suffers from diminishing returns at the chemical signaling level. Therefore, the brain's learning algorithms are tricked into calculating a perpetual positive prediction error. The subconscious mind concludes that this behavior is infinitely better than predicted every single time it occurs. This creates an unstoppable loop of associative learning, The brain becomes completely incapable of updating its internal model to realize that the drug is causing harm. Its neural calculator is locked in a state of permanent delusion, registering an artificial jackpot signal with every administration, driving compulsive seeking behavior despite devastating conscious consequences. The adaptation does not stop at the receptor level. The physical shape of the neurons themselves begins to warp. Neurons communicate via tiny branch-like protrusions called dendrites, which are covered in even smaller structures known as dendritic spines. These spines are the physical landing pads for synaptic connections. Under the influence of chronic dopamine surges and the activation of specific transcription factors within the cell nucleus, the structural anatomy of the nucleus accumbens undergoes a radical physical remodelling. The brain begins to rapidly grow new dendritic spines, specifically tuned to receive inputs from the environment associated with the addiction. This process, known as synaptic plasticity, physically rewires the architecture of the brain. The reward center becomes highly sensitized, growing a massive network of high-speed lanes dedicated solely to processing information related to the addictive substance or behavior. At the same time, the dendritic spines responsible for processing natural rewards, like social interaction, creative achievements, and healthy hobbies, begin to wither away from lack of use. The brain is quite literally sculpting its physical tissue to become incredibly efficient at executing the addiction while structurally dismantling its ability to engage with the rest of reality. This is not a metaphor. It is a physical, anatomical restructuring that locks the behavioral pattern into place. This physical remodeling gives rise to one of the most powerful and destructive phenomena in addiction: automated cue reactivity. As the dense network of new dendritic spines solidifies, it links up with the amygdala, which processes emotion, and the hippocampus, which stores memories. Together they form a hyperreactive cue detection network. This network maps out every single environmental detail associated with the dopamine surges: the street corner where a drug was bought, the sound of an opening app, the time of day, or even an internal state like stress or boredom. Once these neural associations are forged, they become completely independent of the conscious will. When a person in recovery encounters one of these cues, their sensory organs feed the information into the hypersensitized cue reactivity network. Instantly, before the conscious cortex even registers what is happening, the VTA fires a massive, anticipatory burst of dopamine. This is not a spike of pleasure, it is a spike of intense, agonizing craving. The brain registers a sudden, severe deficit between the anticipated reward and the current reality. This anticipatory spike triggers an overwhelming, obsessive drive to close the gap, flooding the individual's consciousness with thoughts of the substance, rendering logical resistance nearly impossible. As the addiction progresses, the locus of neural control undergoes a critical geographical migration within the brain. In the early stages, the choice to engage in the behavior is largely regulated by the ventral striatum, specifically the nucleus accumbens, which evaluates rewards and drives goal-directed behavior. This stage is driven by impulsive desire. However, as the behavior is repeated hundreds or thousands of times under the influence of hyperplastic structural changes, the neural control shifts dorsally. It migrates from the ventral striatum to the dorsal striatum, specifically the putamen and caudate nucleus. The dorsal striatum is the home of your brain's automated habit execution engine. It is the region that allows you to tie your shoes or brush your teeth without consciously thinking about the movements. When an addiction shifts to the dorsal striatum, it transitions from an impulsive choice to a fully automated compulsive habit. The behavior is no longer driven by the expectation of pleasure or reward. It is executed by a deeply buried, unconscious circuit that operates entirely beneath the level of cognitive awareness. The individual can actively hate the behavior, consciously cry out to stop, and experience zero pleasure from the act. Yet their dorsal striatum will execute the motor commands to pursue the substance with flawless, terrifying autonomy. While the deep reward and habit circuits are hyperactivating, they begin to launch a catastrophic assault on the higher-order regions of the brain, the prefrontal cortex. Located directly behind your forehead, the prefrontal cortex is the seat of executive function, rational decision-making, future planning, and impulse control. It acts as the CEO of the brain, sending inhibitory signals down to the primitive subcortical regions to keep raw impulses in check. This is known as top-down control. In a healthy brain, the prefrontal cortex can easily veto a sudden urge generated by the nucleus accumbens. But the massive, chronic dopamine fluctuations characteristic of addiction act like an electrical storm that slowly degrades the connection between the prefrontal cortex and the lower brain. Through a process called hypofrontality, the metabolic activity within the prefrontal cortex drops precipitously. The neurons in this region physically lose their dendritic connections, weakening their ability to fire effectively. The CEO of the brain is essentially forced into a chemical coma. The inhibitory pathways that allow a person to say no to an impulse are structurally eroded. This creates a devastating neurological asymmetry. The bottom-up drive from the hypersensitized habit circuits is stronger than ever, while the top-down brake system of the prefrontal cortex is physically broken. The illusion of addiction is further reinforced by a profound parallel corruption of the brain's internal stress and threat detection machinery. While the dopamine system is downregulating, the extended amygdala, the center for stress, anxiety, and negative affect, is doing the exact opposite. It becomes hyperreactive. In response to the chronic disruption of homeostasis, the hypothalamus begins to secrete high levels of corticotropin-releasing factor, or CRF, which triggers the body's physiological stress response. Concurrently, the brain releases a peptide called dynorphin. Dynorphin acts as the direct chemical opposite of dopamine. It binds to kappa opioid receptors and actively suppresses the reward system, inducing a state of intense dysphoria, restlessness, and psychological pain. When the addictive substance is absent, the individual is not merely returning to a neutral baseline, they are cast into a state of severe neurochemical distress, driven by elevated CRF and dynorphin. The external world feels threatening, overwhelming, and painful. At this stage, the motivation to pursue the addiction shifts completely. It is no longer pursued for the positive reinforcement of a high. It is desperately sought out for negative reinforcement, as a temporary chemical shield to silence the agonizing stress signals generated by their own dysregulated amygdala. We have now arrived at the completion of the cellular trap, where the neuroscience reveals addiction not as a failure of willpower, but as a total structural reorganization of the brain's architecture. The loop is ironclad. We have a ventral tegmental area, and nucleus accumbens that have downregulated their receptors, creating a profound, permanent baseline deficit of joy. We have a hypersensitized cue reactivity network that triggers intense subperceptual cravings at the slightest environmental reminder. We have a dorsal striatum that has locked the behavior into a deeply buried, automated motor habit. We have a prefrontal cortex that has suffered from hypofrontality, leaving its top-down inhibitory brakes completely severed. And finally, we have an extended amygdala flooding the system with stress hormones and dysphoric peptides whenever the substance clears the bloodstream. Every single structural layer of the brain's decision-making and motivational apparatus has been systematically rewritten. The conscious mind becomes a mere spectator, trapped inside a biological machine that has been reprogrammed to seek its own destruction. Understanding this neurobiology is the critical first step, because it shifts the entire conversation away from moral judgment and firmly into the realm of structural neuroscience. This is the foundation upon which the entire pathology of addiction rests. Now that we have uncovered how the midbrain rewards and habits are chemically hijacked, we must look at the executive disaster upstairs. In the next video, we will climb out of the deep reward pathways and examine the literal physical decay of the prefrontal cortex, the brain's CEO, to see exactly why a person can completely understand the logical consequences of their actions, yet remain entirely powerless to stop them. Imagine watching yourself perform an action that you actively, desperately do not want to do. You are not possessed by an outside force, your muscles are moving, your hands are working, and your mind is fully aware of the catastrophic consequences. Yet, you cannot pull the emergency brake. This terrifying disconnection is the hallmark of the executive hijack. In popular culture, addiction is mistakenly framed as a disease of pure hedonism, a relentless pursuit of pleasure. But if you talk to anyone deep in the throes of dependency, they will tell you the pleasure vanished long ago. What remains is a broken steering wheel. The conscious, rational mind becomes a trapped observer inside a biological vehicle it can no longer control. This is not a failure of morals or an absence of willpower. It is the direct, physical degradation of the prefrontal cortex, the outermost layer of the brain responsible for human agency itself. When this executive center is compromised, the brain loses its ability to project into the future, weigh consequences, or veto destructive impulses. The mirror through which the brain observes and regulates itself is shattered, leaving the primitive, short-sighted subcortical circuits to run the entire system into the ground. To understand how this hijack occurs, we must examine the brain's evolutionary hierarchy. The human brain is built in layers, with primitive survival circuits at the bottom and advanced cognitive structures at the top. The apex of this hierarchy is the prefrontal cortex, or PFC. Neuroscientists refer to the PFC as the seat of top-down control. Think of it as a highly sophisticated military command center. It receives raw emotional data from the amygdala and basic drive signals from the striatum. Its job is to analyze this data, compare it against past memories, project the long-term outcomes into the future, and issue inhibitory commands. If the lower brain screams, "I want that food immediately," the healthy PFC steps in and says, "No, that is toxic, wait." This inhibitory control is mediated by long, projecting glutamatergic pathways that travel down from the frontal lobes to the deep subcortical structures. These pathways act as biological brakes, allowing humans to override immediate instincts in favor of long-term survival and social alignment. In a healthy individual, this top-down command structure is absolute. The executive center holds the ultimate veto power over every impulse generated by our ancient biology. Chronic addiction systematically dismantles this command structure through a pathological state called hypofrontality. When the brain is subjected to repeated, artificial dopamine tsunamis, the excessive electrical volatility bleeds into the prefrontal cortex. The delicate neurons of the PFC are not built for this level of chemical turbulence. In response, the brain drastically reduces the baseline metabolic activity and blood flow to the entire frontal lobe. If you look at a functional brain scan of an individual with severe substance or behavioral dependency, the prefrontal cortex does not light up with vibrant activity. It appears dark, quiet, and underfueled. This is hypofrontality, the literal physical silencing of the brain's executive suite. It is the neurological equivalent of a city losing power to its government buildings while the chaotic streets remain fully powered. Because the metabolic engine of the PFC is stalled, the neurons lose their ability to synthesize neurotransmitters efficiently, fire action potentials rapidly, or maintain their complex computational networks. The brain's CEO has not just left the building, it has been starved of the basic resources required to function. Hypofrontality is not just a temporary drop in activity. Over time, it hardens into permanent structural decay. The human brain operates on a strict use-it-or-lose-it principle of neuroplasticity. As the top-down inhibitory pathways from the prefrontal cortex are chronically ignored and bypassed by the hyperactivated habit loops of the lower brain, those frontal pathways begin to wither. Neuroimaging studies consistently reveal a terrifying reality in the brains of addicted individuals, a significant loss of gray matter volume in the prefrontal cortex. Gray matter consists of the neuronal cell bodies and dendrites that process information. When this volume drops, it means neurons are physically shrinking, losing their connections, and in some cases, dying. The physical tissue behind your forehead becomes thinner and less dense. This structural gray matter atrophy directly correlates with the severity and duration of the addiction. It is a profound anatomical truth that explains the progressive nature of the condition. A person cannot simply choose to use willpower to stop, because the physical biological substrate that generates willpower is being systematically disassembled at the cellular level. Within the degraded prefrontal landscape, a specific subregion suffers a catastrophic failure, the orbitofrontal cortex, or OFC. Located right above your eye sockets, the OFC is the brain's internal currency exchanger. It is responsible for calculating the relative value of different options in real time. For example, if you are hungry, the OFC assigns a high value to a sandwich. Once you are full, the OFC rapidly updates its calculations, lowering the value of the sandwich and raising the value of a glass of water. It creates a dynamic value matrix that guides your choices. In addiction, the OFC becomes profoundly dysregulated. The massive dopamine spikes lock the value of the addictive cue at an absolute permanent maximum. The OFC's calculation engine becomes frozen. It assigns an impossibly high value to the drug or behavior, while completely devaluing everything else: career, relationships, financial stability, and physical health. The value matrix is no longer dynamic. It is heavily distorted. The brain looks at a choice between long-term survival and an immediate destructive high, and its broken OFC mathematically concludes that the high is worth more than everything else combined. Directly adjacent to the value matrix lies another critical executive structure, the anterior cingulate cortex, or ACC. The ACC acts as the brain's internal alarm system and error detection filter. It monitors your behavior and compares it to your internal standards and goals. If you make a mistake, slip up on a goal, or do something that compromises your safety, the ACC fires rapidly, generating a distinct neural signal known as error-related negativity. This signal creates a healthy sense of cognitive dissonance and psychological discomfort, forcing you to correct your course. In the executive hijack, the ACC's communication networks are severed. The error detection filter goes completely offline. When an addicted individual engages in a behavior that wrecks their life, the ACC fails to generate the necessary alarm signal. The brain registers the destruction, but fails to feel the internal alarm required to trigger a course correction. The internal warning lights have been unplugged. The individual can clearly see their life falling apart, but the neurological error correction mechanism that normally forces a human to pivot is broken, leaving them to repeat the mistake. The destruction climbs higher into the dorsolateral prefrontal cortex, or DLPFC. This region is the seat of working memory, abstract reasoning, and cognitive flexibility. Cognitive flexibility is the mental capacity to switch between different concepts and adapt your strategy when the environment changes. If a bridge is closed on your way to work, your DLPFC immediately calculates an alternate route. In a brain suffering from executive degradation, the DLPFC loses its plasticity and becomes highly rigid. This manifests as a profound inability to learn from negative consequences. Under normal conditions, if an action leads to pain, arrest, or isolation, the DLPFC updates its behavioral strategy to avoid that action in the future. But an addiction-damaged DLPFC cannot shift its strategy. It becomes locked in a state of cognitive perseveration, meaning it repeats the exact same failing behavior over and over again, expecting a different result. The individual is trapped in a rigid, repetitive loop. entirely incapable of using past negative experiences to formulate a new, safer path forward. A fascinating and tragic consequence of prefrontal degradation is the drastic shrinking of the individual's future time horizon. The prefrontal cortex is the only structure in the known universe capable of mental time travel. It allows humans to project their minds forward by days, months, years, or decades to imagine the long-term outcomes of their current actions. This ability is what drives us to save money, study for exams, and care for our health. When the prefrontal cortex experiences gray matter loss and hypofrontality, this mental time travel mechanism collapses. The future time horizon shrinks from years down to days, then to hours, and finally to minutes. The brain becomes neurologically blind to the future. It enters a state of profound temporal myopia, where only the immediate present exists. If a person cannot neurologically conceptualize the reality of next week, then threats of losing their job, their family, or their life next month hold zero motivational weight. The immediate, short-term drive of the subcortical habit loops wins every single time, because the future has been completely erased from the brain's computational reality. Even as the prefrontal cortex loses its structural integrity and executive control, it does not stop trying to do its secondary job, interpreting the world. The human brain possesses an innate narrative generator, which constantly creates stories to justify our behavior. When the hyperactivated subcortical circuits force a compulsive behavior, the weakened prefrontal cortex cannot block it, but it can rationalize it. This is the neurobiology of denial. The damaged PFC constructs elaborate, highly logical narratives to explain away the self-destruction. It tells the individual, "You've had a stressful day, you deserve this," or "You can stop tomorrow, you are in control." This is a brilliant and insidious survival mechanism of the addiction itself. The brain uses its remaining cognitive intelligence to protect the dependency. The conscious mind genuinely believes these rationalizations because they are being generated by its own executive hardware. The individual is completely blind to the fact that their higher intelligence has been weaponized by their lower biology, turning their rational mind into a defense attorney for their own self-destruction. This rationalization is driven by a fundamental distortion in how the brain attributes meaning to the world, a process known as salience attribution. In a healthy brain, salience, the measure of how important or noticeable something is distributed evenly across life's domains. Food, family, work, and entertainment all hold a healthy share of your attention. This distribution is regulated by prefrontal networks that filter out irrelevant background noise and highlight truly meaningful stimuli. In the executive hijack, these filtering networks suffer a total breakdown. The addictive cue is assigned an absolute monopoly over salience. The brain's attention filters are recalibrated so that anything related to the addiction is hyper-illuminated, while the rest of the world is completely dimmed out. A person walking down the street will instantly spot a subtle cue related to their addiction while failing to notice a friend waving at them. The prefrontal cortex is no longer a neutral judge of reality. It has been transformed into a biased filter that forces the entire conscious experience to revolve around a single destructive focal point. Perhaps the most dangerous aspect of prefrontal degradation is the destruction of the brain's self-awareness networks, a condition known in clinical neurology as anosognosia. Anosognosia means the inability to perceive that you are ill. This is common in stroke patients who cannot move their arm but are genuinely convinced that their arm works perfectly. In severe addiction, a similar phenomenon occurs due to the breakdown of the insular cortex and its connections to the prefrontal lobes. The brain loses its capacity to accurately monitor its own internal state and behavioral patterns. This is why an individual can be in the absolute gutter of life, losing everything, yet look someone in the eye and sincerely say, "I don't have a problem, I can handle it." They are not lying to you, They are literally blind to their own reality. Their internal self-monitoring loop is broken. Without an intact prefrontal network to generate accurate self-awareness, the brain remains completely unaware of its own executive demise, locking the individual inside an invisible cage of biological denial. We have now mapped the complete anatomy of the executive hijack, revealing the profound asymmetry that makes recovery such an immense neurological challenge. The architecture of addiction is incredibly lopsided. The lower, impulsive, habit-driven subcortical structures are running at a massive chemical surplus, reinforced by thousands of repetitions and hyperplastic dendritic spines. Meanwhile, the higher, rational, top-down control structures of the prefrontal cortex are running at a severe structural and metabolic deficit. Expecting an individual in this state to simply decide to stop is a complete misunderstanding of human neurobiology. You are asking a car with a stuck accelerator and entirely vaporized brake pads to stop by sheer force of thought. The executive hijack means that the very tool required to fix the problem, the prefrontal cortex, is the exact tool that is broken. Healing from this state is not about finding a magic life hack or sudden motivation. It requires a slow, calculated, and highly structured environment that protects the brain long enough for the prefrontal gray matter to physically regrow, the value matrix to recalibrate, and the top-down brakes to finally come back online. Now that we have witnessed how the prefrontal cortex is structurally dismantled and its top-down brakes are severed, we must confront the final, deepest layer of the trap. In the next video, we will descend into the very nucleus of the cell to witness the dark side of neuroplasticity, where a permanent molecular switch toggles inside your DNA, giving birth to the anti-reward system and locking the brain into a state of permanent chemical withdrawal. Deep inside the nucleus of your reward neurons lies a silent, microscopic entity that acts as the ultimate point of no return in the architecture of addiction. For decades, scientists believed that neuroplasticity was an exclusively positive force, the brain's wondrous ability to adapt, learn, and heal. But neuroplasticity has a dark, predatory side. When the brain is exposed to chronic, unnatural chemical surges, it doesn't just adapt superficially, it rewires its own genetic expression. A specific, highly stable protein accumulates within the DNA of your nucleus accumbens, acting as a molecular toggle switch. Once this switch is thrown, it alters how your genes are read, locking the brain into a state of structural alteration that can persist for months, years, or even decades after the behavior stops. This is the ultimate trap of addiction. It is a profound shift from a behavioral habit to a permanent molecular restructuring. The brain learns how to be addicted with the exact same flawless biological efficiency it uses to master a musical instrument or a language. Neuroplasticity is not your savior here. It is the concrete that pours into the mold of your dependency, solidifying a destructive pattern directly into your genetic code. To understand this genetic lock, we must look at a transcription factor known to neurobiologists as DeltaFosB. Transcription factors are specialized proteins that bind to specific sequences of DNA, turning genes on or off like light switches. When you experience a normal, everyday pleasure, your brain produces standard proteins that degrade and vanish within a few hours. This ensures your brain returns to a clean baseline. However, addictive stimuli trigger the synthesis of DeltaFosB, an exceptionally stable variant. DeltaFosB possesses a unique atomic structure that resists the brain's natural recycling machinery. Instead of breaking down, it accumulates layer by layer with every single spike of dopamine. Think of it like radioactive sediment building up on the floor of a lake. As the weeks and months of consumption progress, the concentration of DeltaFosB reaches a critical threshold. It begins to permanently alter the transcription of dozens of target genes, fundamentally changing the physical structure of the neuron. This means that even if a person completely stops the behavior, The DeltaFosB remains physically bound to their DNA for an extended period, keeping the addiction machinery fully operational in the background. As DeltaFosB accumulates, it initiates a profound defensive reaction that neuroscientists call the anti-reward system. The brain cannot tolerate being perpetually forced out of homeostatic balance by extreme dopamine spikes. To counter this, it doesn't just lower its dopamine receptors, it constructs an entirely parallel neural network designed to inflict psychological pain. This system is centered in the extended amygdala and the bed nucleus of the stria terminalis. When the brain detects a chronic artificial high, the anti-reward system activates, flooding the synapse with a potent peptide called dynorphin. Dynorphin acts as the literal chemical antithesis to dopamine. While dopamine drives motivation, engagement, and focus, dynorphin induces a state of heavy, suffocating dysphoria, anhedonia, and psychological exhaustion. It binds to kappa opioid receptors on dopamine-releasing neurons, physically shutting down their ability to fire. The birth of the anti-reward system means the baseline of the brain has been severely warped. The natural world is no longer just flat. It is actively painful, because the brain is continuously secreting a chemical designed to make existence feel unbearable. The activation of the anti-reward system is tightly regulated by a molecular cascade involving a protein called CREB. When a massive wave of dopamine hits a neuron, it triggers a rush of cyclic adenosine monophosphate, which activates an enzyme that travels straight into the cell nucleus to stimulate CREB. CREB is the rapid response unit of cellular homeostasis. Its immediate job is to dampen the current high to protect the cell from excitotoxic burnout. It does this by immediately ordering the creation of dynorphin, which loops backward to choke off the dopamine supply. This is the exact neurobiological explanation for acute tolerance, why the second dose of a substance or the second hour of a compulsive behavior never feels as powerful as the first. CREB acts like a heavy blanket thrown over a fire. While CREB degrades relatively quickly when the stimulus stops, its regular activation leaves the door open for the slower, more permanent buildup of DeltaFosB. Together, they create a devastating tag-team dynamic. CREB ruins the pleasure of the immediate high, while DeltaFosB permanently ruins the baseline of everyday life. The anti-reward system doesn't stop at dynorphin. It completely hijacks the body's primary stress network, known as the hypothalamic pituitary adrenal axis, or HPA axis. Under normal conditions, the HPA axis triggers a surge of cortisol and adrenaline when you face a real physical threat, helping you fight or flee. In a brain rewired by DeltaFosB and chronic overstimulation, the threshold for activating this stress axis drops to near zero. The extended amygdala begins to continuously pump out corticotropin-releasing factor, or CRF. This constant chemical alarm keeps the individual in a state of chronic, low-grade physiological panic. Their heart rate variability drops, their muscles tension up, and their mind perceives threats where none exist. Minor daily inconveniences, a traffic jam, a spilled coffee, a subtle change in someone's tone, are interpreted by the hyperreactive HPA axis as life-threatening emergencies. The individual is trapped in an agonizing emotional landscape where their own biology is constantly screaming that they are in immediate danger, driving an overwhelming desperation to find relief. To truly grasp the permanence of this structure, we must look beneath the DNA sequence itself and enter the realm of epigenetics. Your DNA is a vast library of blueprints, but not every blueprint is accessible. To save space, the brain wraps its DNA tightly around tiny spools of protein called histones. If the DNA is wrapped tightly, the gene is silenced. It cannot be read. This is where the dark side of neuroplasticity performs its most insidious trick. The accumulation of DeltaFosB triggers a process called histone methylation and acetylation within the reward center. It physically forces the histones to unwind around specific genes that promote addiction, making them permanently easy to read. Simultaneously, it tightly locks up the histones around the genes responsible for growth, learning, and emotional resilience. This is an epigenetic landscape shift. The structural code of your identity has not changed, but the access permissions have been radically modified. The brain has been epigenetically reprogrammed to keep the gates of craving wide open, while sealing shut the pathways of self-regulation and natural satisfaction. This epigenetic restructuring directly targets one of the most vital growth factors in the human nervous system, brain-derived neurotrophic factor, or BDNF. BDNF is often described as fertilizer for the brain. It is essential for neurogenesis, synaptic health, and the formation of new adaptive memories. In a healthy brain, BDNF allows you to learn from your mistakes and build a resilient mind. However, the DeltaFosB switch subverts this system entirely. It forces a massive upregulation of BDNF within the specific pathway that connects the ventral tegmental area to the nucleus accumbens. Instead of fertilizing the whole brain, this precious growth factor is concentrated entirely into feeding the addiction loop. It accelerates the growth of the pathological dendritic spines we discussed in the first video, making the craving pathways incredibly robust and physically massive. Meanwhile, BDNF levels in the prefrontal cortex plummet, leaving the rational brain starving for growth factors and unable to repair its damaged gray matter. The brain's natural healing system has been completely weaponized against itself. The molecular chaos eventually spills out of the dopamine system and completely corrupts the brain's primary excitatory neurotransmitter, glutamate. Glutamate is the chemical currency of thought, learning, and communication across the entire cortex. In a healthy brain, glutamate levels are kept under strict regulation by specialized cleanup cells called astrocytes. These astrocytes ensure that glutamate signals are crisp, clean, and temporary. In the advanced stages of addiction, the genetic changes driven by DeltaFosB cause a dramatic downregulation of the primary glutamate transporter, GLT1, in the prefrontal accumbens pathways lose their ability to clear away excess glutamate. This results in a permanent state of leaky glutamate homeostasis. The reward center is subjected to a constant, chaotic background whisper of excitatory noise. Because the signaling is no longer clean, the prefrontal cortex completely loses its ability to send clear, crisp instructions to the lower brain. The lines of communication are filled with static, rendering any conscious effort to direct focus or control behavior completely unintelligible to the deep reward hardware. When you combine receptor downregulation, the dynorphin anti-reward system, hyperreactive CRF, and leaky glutamate homeostasis, the brain sinks into a profound state known as the anhedonic void. Anhedonia is not just feeling sad, it is the absolute structural inability to experience pleasure from any natural stimulus. Food tastes like cardboard, music sounds like random noise, social connections feel exhausting and hollow. This void is a direct consequence of a deeply depressed baseline reward current, because the brain's internal reward currency has been hyperinflated by artificial surges. The subtle, beautiful pulses of dopamine generated by normal human life are completely beneath the detection threshold of the remaining desensitized receptors. The individual is marooned in a desolate psychological landscape where the color has been completely drained from reality. At this stage, the pursuit of the addictive behavior is no longer motivated by a desire to feel good. It is a desperate, frantic attempt to simply crawl out of the anhedonic void and return to a fleeting moment of neutral baseline comfort. As the molecular pathology deepens, it triggers an immune response within the brain itself, introducing the destructive element of chronic neuroinflammation. The brain possesses its own dedicated immune cells called microglia. For a long time, microglia were thought to only respond to physical trauma or bacterial infections. However, recent neuroscience reveals that chronic, high-dose dopamine volatility and the systematic breakdown of glutamate homeostasis act as a profound metabolic stressor that alerts these immune guards. The microglia shift from a calm, protective state into a hyperaggressive, pro-inflammatory phenotype. They begin to release a cascade of inflammatory proteins called cytokines, such as tumor necrosis factor alpha and interleukin-1 beta, directly into the reward pathways in the prefrontal cortex. This neuroinflammation behaves exactly like a hidden, low-grade fever burning through your neural networks. It impairs synaptic plasticity, slows down cellular metabolism, and accelerates gray matter atrophy. The individual is not just dealing with a psychological habit, their brain tissue is experiencing a physical, inflammatory illness that actively degrades their cognitive and emotional capacity. The revelation of this deep molecular, genetic, and inflammatory architecture exposes the complete absurdity of the popular cultural myth surrounding addiction, the myth of the quick fix. We are bombarded with pseudoscientific hacks, motivational advice, and simplistic mindset shifts that promise to rewire your brain in 21 days. The rigid reality of structural neuroscience completely obliterates these claims. You cannot think your way out of an epigenetic modification. You cannot use pure willpower to instantly degrade accumulated DeltaFosB proteins that are physically bound to your DNA. You cannot use a motivational quote to force your astrocytes to suddenly rebuild their lost GLT-1 glutamate transporters, or command your hyperaggressive microglia to stop releasing inflammatory cytokines. Addiction is a systemic, structural, and genetic reorganization of the brain's survival machinery. It is a biological trap designed by evolution to be incredibly difficult to escape. Acknowledging the sheer depth and complexity of this cellular reality is not a message of despair. It is a vital injection of scientific truth that frees us from the cycle of shame and useless, short-term solutions. How, then, does the brain ever escape this cellular trap? The answer lies in understanding that just as the dark side of neuroplasticity built the prison, a slow, highly calculated path of molecular reversion can dismantle it. Because DeltaFosB is incredibly stable, it requires an extended, uninterrupted window of time without the artificial dopamine spikes to naturally decay and detach from the DNA. This is not a matter of weeks, it is a biological timeline of many months. As the DeltaFosB levels slowly recede, the epigenetic landscape begins to shift back. The histones allow access to the silenced genes once more. The astrocytes slowly rebuild their GLT-1 transporters, stabilizing the glutamate whispers. The microglia calm their inflammatory response, and the anti-reward system gradually lowers its production of dynorphin and CRF. The baseline of joy slowly rises back toward zero. This is a physical, anatomical healing process that mirrors the slow knitting together of a fractured bone. It cannot be rushed, and it cannot be short-circuited. By understanding the deep neuroscience of this path, we finally see addiction for what it truly is, a profound structural challenge that requires a precise biological timeline to heal. If you prefer objective, data-driven neuroscience explanations over superficial self-help hacks, you've found your community. Subscribe to keep building your neurological literacy. This is Archive of Becoming, and I will see you in the next deep dive.